Hi, I am Dr. Anshul Garg with interesting and learning episode from one of the important units of Biotechnology, which is prepared by Dr. Rupal Dubey, Assistant Professor, Guest Faculty, Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur, Central University, Sagar, Madhya Pradesh, India. In this episode, we are going to discuss about enzyme immobilization. The current demands of sustainable green methodologies have increased the use of enzymatic technology in industrial processes. Employment of enzyme as biocatalyst offers the benefits of mild reaction conditions, biodegradability, and catalytic efficiency. The harsh conditions of industrial processes, however, increase propensity of enzyme destabilization, shortening their industrial lifespan. Consequently, the technology of enzyme immobilization provides an effective means to circumvent these concerns by enhancing enzyme catalytic properties and also simplify downstream processing and improve operational stability. There are several techniques used to immobilize the enzymes onto supports, which range from reversible physical adsorption and ionic linkages to the irreversible stable covalent bonds. Such techniques produce immobilized enzymes of varying stability due to changes in the surface microenvironment and degree of multi-point attachment. In this episode, we are going to discuss all aspects related to enzyme immobilization. Let's start our episode while taking a look at what we are going to discuss today. The lecture has been divided into 5 modules. Module 1 is Introduction to Enzyme Immobilization. Module 2 is about technique of enzyme immobilization. Module 3 is about factors affecting enzyme kinetics. Module 4 is about immobilization of bacteria and plant cells. And Module 5 is about study of different enzymes. Let's start with Module 1, which is about Introduction to Enzyme Immobilization. The term immobilized enzymes refers to enzymes physically confined or localized in a certain defined region of space with retention of their catalytic activities and which can be used repeatedly and continuously. Besides the application in industrial processes, the immobilization techniques are the basis for making a number of biotechnological products with applications in diagnostics, bioaffinity, chromatography, and biosensors. Initially, only immobilized single enzymes were used, but the 1970s saw the development of more complex systems, including 2 enzyme reactions with cofactor regeneration and living cells. The major components of an immobilized enzyme system are the enzyme, the matrix, and the mode of attachment. The enzymes can be attached to the support by interactions ranging from reversible physical adsorption and ionic linkages to stable covalent bonds. Thus, immobilization of enzymes or cells refers to the technique of confining or anchoring the enzymes or cells in or on an inert support for their stability and functional reuse. By employing this technique, enzymes are made more efficient and cost effective for their industrial use. Some workers regard immobilization as a goose with a golden egg in enzyme technology. Immobilized enzymes retain their structural conformation necessary for catalysis. History of enzyme immobilization. It is possible to visualize 3 steps in the development of this immobilized biocatalysts, that is given in table 1. In the first step, at the beginning of the 19th century, immobilized microorganisms were being employed industrially on an empirical basis. This was the case with both the microbial production of vinegar by letting alcohol containing solution trickle over wood shavings overgrown with bacteria and the development of the trickling filter or percolating process for wastewater clarification. The modern history of enzyme immobilization goes back to the late 1940s, but much of the early work was largely ignored by biochemists because it was primarily published in journals of other disciplines. The basis of the present technologies was developed in the 1960s and there was an explosive increase in publications. In the second step, only immobilized single enzymes were used, but by the 1970s, more complex systems including two enzyme reactions with cofactor regeneration and living cells were developed. As an example of the latter, we can mention the production L amino acids from alpha keto acids by stereoselective reductive amination with L amino acid dehydrogenase. The process involves the consume of nicotinamide adenine dinucleotide, that is NADH, and regeneration of the coenzyme by coupling the amination with the enzymatic oxidation of formic acid to carbon dioxide with concomitant reduction of NAD plus to NADH in the reaction catalyzed by the second enzyme formate dehydrogenase. Now, table 1 shows steps in the development of immobilized enzymes. Step first, date is 1815. Empirical use in processes such as acetic acid and wastewater treatment. Step second occurred in 1960s. Single enzyme immobilization, production of single enzyme immobilization, production of L amino acids, isomerization of glucose. Step third, 1985 to 1995. Multiple enzyme immobilization including cofactor regeneration and cell immobilization. Example, production of L amino acids from keto acids in membrane reactor. Salient features of enzyme immobilization. The enzyme phase is called as carrier phase, which is water insoluble but hydrophilic porous polymeric matrix. Example is agarose, cellulose, etc. The enzyme phase may be in the form of fine particulate, membranous or microcapsule. The enzyme in turn may be bound to another enzyme via cross-linking. A special module is produced employing immobilization techniques through which fluid can pass easily, transforming substrate into product and at the same time facilitating the easy removal of catalyst from the product as it leaves the reactor. The support or carrier utilized in immobilization technique is not stable at particular pH, ionic strength or solvent conditions. Hence, may be disrupted or dissolved, releasing the enzyme component after the reaction. Advantages of enzyme immobilization. Multiple or repetitive use of a single batch of enzymes. Immobilized enzymes are usually more stable. Ability to stop the reaction rapidly by removing the enzyme from the reaction solution. Product is not contaminated with the enzyme. Easy separation of the enzyme from the product. It allows development of a multi-enzyme reaction system. Reduces effluent disposal problems. Stable and more efficient in function. Can be reused again and again. Products are enzyme free. Ideal for multi-enzyme reaction systems. Control of enzyme function is easy. Suitable for industrial and medical use. Minimize effluent disposal problems. There are, however, certain disadvantages also associated with immobilization, such as the possibility of loss of biological activity of an enzyme during immobilization or while it is in use. Immobilized enzymes are generally preferred over immobilized cells due to specificity to yield the products in pure form. However, there are several advantages of using immobilized multi-enzyme systems such as organelles and whole cells over immobilized enzymes. The immobilized cells possess the natural environment with cofactor availability and also its regeneration capability and are particularly suitable for multiple enzymatic reactions. Now, coming on to disadvantages of enzyme immobilization. It gives rise to an additional bearing on cost, invariably affects the stability and activity of enzymes. The technique may not prove to be of any advantage when one of the substrate is found to be insoluble. Certain immobilization protocols offer serious problems with respect to the diffusion of the substrate to have an access to the enzyme. Choice of matrix. The terms solid phase support, carrier and matrix are used synonymously. The characteristics of the matrix are of paramount importance in determining the performance of the immobilized enzyme system. Ideal support properties includes physical resistance to compression, hydrophilicity, inertness towards enzymes, ease of derivatization, biocompatibility, resistance to microbial attack, and availability at low cost. Supports can be classified as inorganic and organic according to their chemical composition as given in table 2. The organic supports can be subdivided into natural and synthetic polymers. The physical characteristics of the matrices such as mean particle diameter, swelling behavior, mechanical strength and compression behavior will be of major importance for the performance of the immobilized systems and will determine the type of reactor used under technical conditions, that is stirred tank, fluidized or fixed beds. In particular, pore parameters and particle size determine the total surface area and thus critically affect the capacity for binding of enzymes. Non-porous supports show few diffusional limitations but have a low loading capacity. Therefore, porous supports are generally preferred because the high surface area allows for a higher enzyme loading and the immobilized enzyme receives greater protection from the environment. Porous supports should have a controlled pore distribution in order to optimize capacity and flow properties. In spite of the many advantages of inorganic carriers, for example, high stability against physical, chemical and microbial degradation, most of the industrial applications are performed with organic matrices. The hydrophilic character is one of the most important factors determining the level of activity of an immobilized enzyme. An excellent matrix that has been extensively used is agarose. In addition to its high porosity, which leads to a high capacity for proteins, some other advantages of using agarose as a matrix are hydrophilic character, ease of derivatization, absence of charge groups which prevents non-specific adsorption of substrate and products, and commercial availability. However, an important limitation in the use of agarose and other porous supports is the high cost. This problem can be circumvented by employing reversible methods that allow matrix regeneration and reuse. Table 2 shows classification of matrix as we discussed earlier. Matrix is classified into two types: organic natural polymers, which includes polysaccharides such as cellulose, dextrans, agar, agarose, chitin, alginate; proteins, which include collagen or albumin and carbon. Synthetic polymers include polystyrene and other polymers such as polyacrylate, polymethacrylate, polyacrylamide, polyamides, vinyls and allyl polymers. Inorganic polymers include natural minerals such as bentonite and silica, processed materials such as glass, both non-porous and controlled pore, metals, controlled pore metal oxides, etc. Now let's start with module 2, technique of enzyme immobilization. The enzymes can be attached to the support via interactions ranging from reversible physical adsorption and ionic linkages to stable covalent bonds. One way of classifying the various approaches to immobilizing enzymes is in two broad categories: irreversible and reversible methods. The strength of the binding is usually inversely related to the ease with which it can be reversed. These two conflicting objectives, that is stability and reversibility, are difficult to fulfill simultaneously. The traditional approach has been to make the bond as strong as possible and sacrifice reversibility. Now coming on to methods of irreversible enzyme immobilization. The concept of irreversible immobilization means that once the biocatalyst is attached to the support, it cannot be detached without destroying either the biological activity of the enzyme or the support. The most common procedures of irreversible enzyme immobilization are covalent coupling, entrapment or microencapsulation and cross-linking as shown in figure 1. Now coming on to formation of covalent bonds. Immobilization of proteins by methods based on the formation of covalent bonds are among the most widely used. An advantage of these methods is that because of the stable nature of the bonds formed between enzyme and matrix, the enzyme is not released into the solution upon use. However, in order to achieve high levels of bound activity, the amino acid residues essential for catalytic activity must not be involved in the covalent linkage to the support. This may prove a difficult requirement to fulfill in some cases. A simple procedure that sometimes improves the activity yield is to carry out the coupling reaction in the presence of substrate analogues. Covalent methods for immobilization are employed when there is a strict requirement for the absence of the enzyme in the product. A wide variety of reactions have been developed depending on the functional groups available on the matrix. Coupling methods in general can be divided into two main classes. First is activation of the matrix by addition of a reactive function to a polymer. Second, modification of the polymer backbone to produce an activated group. The activation processes are generally designed to generate electrophilic groups on the support, which in the coupling step react with the strong nucleophiles on the proteins. The basic principles controlling the course of covalent coupling to the matrices are analogous to those used for the chemical modification of proteins. The most frequently used reactions involve the following side chains of the amino acids: lysine, E amino group, cysteine, thiol group, and aspartic and glutamic acids of carboxylic group. The covalent reactions commonly employed give rise to enzymes linked to the support through either amide, ether, thioether, or carbamate bonds. And in many cases, it is also stabilized. Figure 1 shows approaches to enzyme immobilization. Advantages of covalent coupling. The strength of the binding is very strong, so leakage of enzyme from the support is absent or very little. This is a simple, mild, and often successful method of wide applicability. Now coming on to disadvantages of covalent coupling. Enzymes are chemically modified and so many are denatured during immobilization. Only small amounts of enzymes may be immobilized, about 0.02 grams per gram of matrix. Now coming on to second method, that is entrapment. The entrapment method is based on the occlusion of an enzyme within a polymeric network that allows the substrate and the products to pass through but retain the enzyme. This method differs from the coupling methods described above in that the enzyme is not bound to the matrix or membrane. There are different approaches to entrapping enzymes such as gel or fiber entrapping and microencapsulation. The practical use of these methods is limited by mass transfer limitations through membranes or gels. Entrapment is carried out by mixing the biocatalyst into a monomer solution, followed by polymerization initiated by a change in temperature or by a chemical reaction. Polymers like polyacrylamide, collagen, cellulose acetate, calcium alginate or carrageenan, etc. are used as the matrices. Enzymes can be entrapped by several ways. First is enzyme inclusion in gels. This is an entrapment of enzymes inside the gels. Second is enzyme inclusion in fibers. The enzymes are trapped in a fiber format of the matrix. Enzyme inclusion in microcapsules. In this case, the enzymes are trapped inside a microcapsule matrix. The hydrophobic and hydrophilic forms of the matrix polymerize to form a microcapsule containing enzyme molecules inside. The major limitation for entrapment of enzymes is the leakage from the matrix. Most workers prefer to use the technique of entrapment for immobilization of whole cells. Entrapped cells are in use for industrial production of amino acids such as L-isoleucine, L-aspartic acid, L-malic acid and hydroquinone. Advantages of entrapment. Loss of enzyme activity upon immobilization is minimized. Disadvantages of entrapment. The enzyme can leak into the surrounding medium. Another problem is the mass transfer resistance to substrates and products. Substrate cannot diffuse deep into the gel matrix. Now coming on to methods of reversible immobilization. Because of the type of the enzyme support binding, reversibly immobilized enzymes can be detached from the support under gentle conditions. See figure 2. The use of reversible methods for enzyme immobilization is highly attractive, mostly for economic reasons, because when the enzymatic activity decays, the support can be regenerated and reloaded with fresh enzyme. Indeed, the cost of the support is often a primary factor in the overall cost of immobilized catalyst. The reversible immobilization of enzymes is particularly important for immobilizing labile enzymes and for applications in bioanalytical systems. Now coming on to next method, which is adsorption through non-covalent interactions. Non-specific adsorption. The simplest immobilization method is non-specific adsorption, which is mainly based on physical adsorption or ionic binding. In physical adsorption, the enzymes are attached to the matrix through hydrogen bonding, van der Waals forces or hydrophobic interactions, whereas in ionic bonding, the enzymes are bound through salt linkages. The nature of the forces involved in non-covalent immobilization results in a process that can be reversed by changing the conditions that influence the strength of the interaction. For example, pH, ionic strength, temperature or polarity of the solvent. Immobilization by adsorption is a mild, easy to perform process and usually preserves the catalytic activity of the enzyme, such as enzyme leakage from matrix when the interactions are relatively weak. Next method is ionic binding. An obvious approach to the reversible immobilization of enzymes is to base the protein-ligand interactions on principles used in chromatography. For example, one of the first applications of chromatographic principles in the reversible immobilization of enzymes was the use of ion exchangers. The method is simple and reversible, but in general it is difficult to find conditions under which the enzyme remains both strongly bound and fully active. More recently, the use of immobilized polymeric ionic ligands has allowed for modulation of protein matrix interactions and has thus optimized the properties of the derivative. A number of patents have been filed on the use of polyethyleneamine to bind a rich variety of enzymes and whole cells. However, problems may arise from the use of a highly charged support when the substrates or products themselves are charged. The kinetics are distorted as a result of partition or diffusion phenomena. Therefore, enzyme properties such as pH optimum or pH stability may change. Although this could pose a problem, it could also be useful to shift the optimal conditions of a certain enzyme towards more alkaline or acidic conditions depending on the application. Next is hydrophobic adsorption. Another approach is the use of hydrophobic interactions. In this method, it is not the formation of chemical bonds, but rather an entropically driven interaction that makes place. Hydrophobic adsorption has been used as a chromatographic principle for more than 3 decades. It relies on well-known experimental variables such as pH, salt concentration, and temperature. The strength of interaction relies on both the hydrophobicity of the adsorbent and the protein. The hydrophobicity of the adsorbent can be regulated by the degree of substitution of the support and by the size of the hydrophobic ligand molecule. The successful reversible immobilization of beta amylase and amyloglucosidase to hexyl agarose carriers has been reported. Two hydrophobic adsorbents have also been reported. Next is affinity binding. The principle of affinity between complementary biomolecules has been applied to enzyme immobilization. The remarkable selectivity of the interaction is a major benefit of the method. However, the procedure often requires the covalent binding of a costly affinity ligand, example antibody or lectin, to the matrix. Approaches to enzyme immobilization is shown in figure 2 through reversible methods. Now coming on to advantages of adsorption. Little or no conformation change of the enzyme, simple and cheap, no reagents are required, wide applicability and capable of high enzyme loading. Now talking about disadvantages of adsorption. Desorption of the enzyme protein resulting from changes in temperature or pH and ionic strength. It is a slow method. Now coming on to chelation or metal binding method. Transition metal salts or hydroxides deposited on the surface of organic carriers become bound by coordination with nucleophilic groups on the matrix. Mainly titanium and zirconium salts have been used and the method is known as metal link immobilization. The metal salt or hydroxide is precipitated onto the support, for example cellulose, chitin, alginic acid or silica based carriers, by heating or neutralization. Because of steric factors, it is impossible for the matrix to occupy all coordination positions of the metal. Therefore, some of the positions remain free to coordinate with groups from the enzymes. The method is quite simple, and the immobilized specific activities obtained with enzymes in this way have been relatively high, that is 30 to 80%. However, the operational stabilities achieved are highly variable and the results are not easily reproducible. The reason for this lack of reproducibility is probably related to the existence of non-uniform adsorption sites and to a significant metal ion leakage from the support. In order to improve the control of the formation of the adsorption sites, chelator ligands can be immobilized on the solid supports by means of stable covalent bonds. The metal ions are then bound by coordination and the stable complexes formed can be used for the retention of proteins. Elution of the bound proteins can be easily achieved by competition with soluble ligands or by decreasing pH. The support is subsequently regenerated by washing with a strong chelator such as ethylene diamine tetraacetic acid, that is EDTA, when desired. These metal chelated supports were named IMA immobilized metal ion affinity adsorbents and have been used extensively in protein chromatography. The approach of using different IMA gels as supports for enzyme immobilization has been studied using Escherichia coli beta-galactosidase as a model. Now next method is formation of disulfide bonds. These methods are unique because even though a stable covalent bond is formed between matrix and enzyme, it can be broken by reaction with a suitable agent such as dithiothretol, that is DTT, under mild conditions. Additionally, because the reactivity of the thiol groups can be modulated via pH alteration, the activity yield of the methods involving disulfide bond formation is usually high, provided that an appropriate thiol reactive adsorbent with high specificity is used. Next method is microencapsulation. Microencapsulation is a type of entrapment. It refers to the process of spherical particle formation wherein a liquid or suspension is enclosed in a semi-permeable membrane. The membrane may be polymeric, lipoidal, lipoprotein based or non-ionic in nature. There are three distinct ways of microencapsulation. First, building of special membrane reactors. Second, formation of emulsions. Third, stabilization of emulsions to form microcapsules. Fourth, microencapsulation is recently being used for immobilization of enzymes and mammalian cells. For instance, pancreatic cells grown in cultures can be immobilized by microencapsulation. Hybridoma cells have also been immobilized successfully by this technique. Next method is cross-linking. The absence of a solid support is a characteristic feature of immobilization of enzymes by cross-linking. The enzyme molecules are immobilized by creating cross links between them through the involvement of polyfunctional reagents. These reagents, in fact, react with the enzyme molecules and create bridges which form the backbone to hold enzyme molecules. There are several reagents in use for cross-linking. These include glutarialdehyde, diazobenzidine, hexamethylene diisocyanate, and toluene diisothiocyanate. Glutarialdehyde is the most extensively used cross-linking reagent. It reacts with lysyl residues of the enzyme and forms a Schiff's base. Cross links formed between the enzyme and glutarialdehyde are irreversible and can withstand extreme pH and temperature. Glutarialdehyde cross-linking has been successfully used to immobilize several industrial enzymes, for example, glucose isomerase, penicillin amidase, etc. The technique of cross-linking is quite simple and cost-effective, but the disadvantage is that it involves the risk of denaturation of the enzyme by the polyfunctional reagent. Module 3 is about factors affecting enzyme kinetics. Coming on to choice of immobilization technique. The selection of a particular method for immobilization of enzymes is based on a trial and error approach to choose the ideal one. Among the factors that decide a technique, the enzyme catalytic activity, stability, regenerability and cost factor are important. Immobilization of L-amino acid acylase. L-amino acid acylase was the first enzyme to be immobilized by a group of Japanese workers, Shibata and Tosa, 1969. More than 40 different immobilization methods were attempted by this group. Only 3 of them were found to be useful. They were covalent binding to iodoacetyl cellulose, ionic binding to DEAE, Cefadex, and entrapment within polyacrylamide. Stabilization of soluble enzymes. Some of the enzymes cannot be immobilized and they have to be used in soluble form. For example, enzymes used in liquid detergents, some diagnostic reagents, and food additives. Such enzymes can be stabilized by using certain additives or by chemical modifications. The stabilized enzymes have longer half-lives, although they cannot be recycled. Some important methods of enzyme stabilization are briefly described. First is solvent stabilization. Certain solvents at low concentrations stabilize the enzymes, while at high concentrations the enzymes get denatured. For example, acetone 5% and ethanol 5% can stabilize benzyl alcohol dehydrogenase. Substrate stabilization. The active site of an enzyme can be stabilized by adding substrates. For example, starch stabilizes A-amylase. Glucose stabilizes glucose isomerase. Next is stabilization by polymers. Enzymes can be stabilized particularly against increased temperature by addition of polymers such as gelatin, albumin and polyethylene glycol. Stabilization by salts. Stability of metalloenzymes can be achieved by adding salts such as calcium, ferrous, manganese, copper and zinc. Example, proteases can be stabilized by adding calcium. Stabilization by chemical modifications. Enzymes can be stabilized by suitable chemical modifications without loss of biological activity. There are several types of chemical modifications. First is addition of polyamino side chains, for example polytyrosine and polyglycine. Acylation of enzymes by adding groups such as acetyl, propionyl and succinyl. Stabilization by rebuilding. Theoretically, the stability of the enzymes is due to hydrophobic interactions in the core of the enzyme. It is therefore proposed that enzymes can be stabilized by enhancing hydrophobic interactions. For this purpose, the enzyme is first unfolded and then rebuilt in one of the following ways. First, the enzyme can be chemically treated, for example, as in urea and a disulfide, and then refolded. The refolding can be done in the presence of low molecular weight ligands. For certain enzymes, refolding at higher temperatures, such as around 50 degrees Celsius, stabilize them. Stabilization by site-directed mutagenesis. Site-directed mutagenesis has been successfully used to produce more stable and functionally more efficient enzymes, for example, subtilisin E.